Anthrax is a zoonotic, viral, and contagious disease caused by gram-positive spore-forming bacteria. This bacteria is called Bacillus anthracis. Anthrax was originally found in sheep during the 18th century as an epidemic destroyed about half of the sheep population in Europe. In 1876, Robert Koch studied Bacillus anthracis and found that the bacteria could form spores that were viable for long periods of time. In 1877, Koch grew this organism and planted it into healthy animals where he found that it produced the infection known as anthrax. Anthrax was the first bacterial infection to be solved using the idea of Koch's postulates. During the first half of the 20th century, 20,000 to 100,000 human cases of anthrax occurred on a yearly basis worldwide. During the second half of the 20th century, this number decreased to 2,000 cases.
When dealing with anthrax, this is a bacteria which form an infection is through spores. When released into the air, this activates the bacteria to produce a toxin which, left untreated, leads to illness. Anthrax can infect the host through breathing the spores, eating or drinking water that is contaminated with spores, getting spores into a cut or scrape in the skin, eating raw or uncooked meat from infected animals. This is rarely reported due to regulations on vaccination for animals. With recent data, it was also shown that there is another type of infection, which is injecting anthrax. This involves people that inject heroin. People that are in environments with animals or work with animals are more susceptible to being exposed to anthrax. This includes being around infected animals or working with animal products like wool, hide, or hair.
The most well-known cases of anthrax bioterrorism attacks occurred in the United States in late 2001, soon after the 9/11 terrorist attacks. 5 Americans were killed and 17 were sickened. This became the worst biological attack in U.S. history. The anonymous letters laced with deadly anthrax spores arrived at media companies and congressional offices, including Senate Majority Leader Tom Daschle's. Physicians were told to be on high alert for anthrax cases, and these cases contributed to a national sense of distress. This outbreak resulted in the formation of the Anthrax Task Force, which consisted of FBI investigators and other agencies. The case also encouraged new scientific breakthroughs in determining more about anthrax, including researchers at Wisconsin and Harvard identifying the bacteria's lethal receptor. In July of 2008, Bruce Ivins, who was suspected in the 2001 anthrax attacks, committed suicide. The U.S. Attorney made a statement that Ivins was responsible for the attacks, but there still remains the possibility that someone else may have been involved as well. And a fun fact is Netflix recently released a documentary called The Anthrax Attacks in the Shadow of 9/11 that has brought even more attention to this. Anthrax, the bacterial cellular pathogen, is caused by the aerobic, spore-forming, gram-positive rod-shaped bacterium Bacillus anthracis. Anthrax's major virulence factors are the anthrax toxins and an anti-phagocytic polyglutamic capsule. These are encoded by 2 large plasmids, PX01 and PX02. The plasmid's expression is controlled by the bicarbonate responsive transcriptional regulator ATXA.
One of the virulence factors, the anthrax toxins, consists of 3 polypeptides, the protective antigens, lethal factor, and edema factor, which come together to form lethal and edema toxins. The protective antigens bind to receptors to translocate the lethal factors and edema factors into cells. The toxins then alter signal pathways into the host to interfere with innate immune responses in the early stages of infection and consequently induce vascular collapse at late stages.
The first vaccine made for anthrax contained attenuated live organisms and was produced in 1881 by Louis Pasteur. He tested this vaccine on a group of 50 cows, where 25 received the vaccine and 25 received the virulent form of anthrax. The 25 vaccinated cows survived, but the others did not. The first human case of inhalation anthrax occurred during the early 1900s among workers in both textile and tanning industries, where goat skin, goat hair, and wool was processed. In 1950, the Army Chemical Corps developed the first human anthrax vaccine, where it was 92.5% effective. By 1970, it was replaced with an updated and licensed version of the vaccine. In order to diagnose anthrax, a chest X-ray or a CT scan need to be taken. Symptoms of anthrax are nonspecific, but usually consist of a fever, fatigue, a cough, general malaise, myalgias, and even chest pains. A chest X-ray can reveal upper or lower lobe infiltrates, pleural effusions, or mediastinal widening, while a CT scan can define mediastinal lymphadenopathy. Taking a nasal swab in an attempt to grow Bacillus anthracis or by testing the blood directly can lead to a diagnosis of anthrax. The treatment for anthrax includes various intravenous combination antibiotic therapy.